Hello and welcome to another Wolfram Institute live stream. It's been a while. Today we have Matheo Tripnaux. I hope I pronounced that correctly. He'll be talking about his the Wolfram Summer Research Institute project called Self-Replication for rewriting systems.
Yes,
Matteo,
thank
take it
you.
away. Go ahead.
So yes, hello everyone. I'm going to talk about substrate agnostic definition of self-replication in rewriting systems. I'm going to try to assume zero background knowledge and to explain everything from scratch so that everyone can understand it properly. It's fortunate because these concepts are kind of intuitive, so I think it will work very easily. So yes, I started doing this project during the Wolfram summer school, and then I developed it further during the past few weeks. And now I'm trying to turn it into an academic paper and to work further on it, to develop it into an actual framework. So first, let's discuss why self-replication is important. So the basic idea of self-replication is that you copy yourself into the exact same thing you are. You can be anything from a small molecule to a huge computer system. And the idea is that the copy is exact. So it's different from reproduction that we observe in living beings mostly. And the difference is that there is no random mutation. There is no modification. The copy is exact. So in biology, we're mostly interested in reproduction because this is the support for natural selection and evolution in general. But there is one specific field of biology where self-replication is especially relevant, which is called the origin of life. And the goal of this field is to find what's in between the soup of chemicals that were there before life and the first free cell that then starts mutating into what we know, humanity and so on. So this question is very important, and especially we believe that the organisms were started with self-replication, and then the mutation arose and everything started to evolve properly as we know it. So this is a key problem, especially understanding the minimal conditions for self-replication and trying to define it properly so that we can test for it, we can understand it correctly, and we can run experiments. Then what is a rewriting system? Because that's the end of the title. Well, it's very simple. You select an initial state. You can choose the shape of the state, it doesn't matter. And then you define some rewrite rules on it, which means that it's some rules that depend only on the state and some environmental parameters if you want. And then these rules will change the state in the next step of the system. So let's have a look at some examples. You have the string substitution system, or you have a sequence of characters, such as, I don't know, A and B, for instance. And here you have some rules. You have A that turns into AB and B that turns into A. And if you run the program on an initial condition that is just A, you get this growing structure, which can have interesting patterns and so on. You have there's plenty of examples, but one of them is the L systems. It's a subset of string substitution that has some geometric properties that are binded to them. And in that case, these very small rule can produce a fractal, which is a very fancy picture, which has very good mathematical properties and so on. You also have this other substrate, this other rewriting system that is called graph rewriting, or hypergraph, or Wolfram model. The idea is that you have this shape here on the left-hand side, and then whenever you detect it inside of a graph, you will rewrite it into the right-hand side of the rule. And if you do that a lot of time, you can have these complex structures that emerge and that are extremely relevant to lots of things, society, physics, and so on. Then we have cellular automata that I'm sure every one of you knows about. You've got this specific one that is called game of life, where you have these rules and you apply it to 2D grid, and then it gets complicated very quickly. And this is a famous example of a glider. It's something that seems like it moves in a direction, even though it's only based on simple rules. So these are examples of rewriting systems. And there is many more. And now, what is self-replication in that context? Well, the intuition is that something produces itself when we apply some set of the rules, the rewriting rules, to this thing multiple times, or like any given amount of time. So for instance, if I'm in the position of an initial condition that is A, and I apply my rule that is A turns into A and A, I will have two copies of A. So this is self-replication because I replicated the A into different A's. And in the next step, it will grow and grow again. But this is not mandatory. It is not supposed to have an exponential growth. But this is something interesting as well. The only requirement is that we do not want the state to be periodic or a fixed point, which means that we do not want the self-replicating candidate to be just something that exists and stays, because otherwise it's just persisting and existing and not exactly replicating. It's just creating a copy of itself in the next time step, right? And this is an example of a periodic thing in Game of Life, which is called a jam. It basically has a cycle of 3 and it loops back to himself. So it's not replication. This is the intuition we have. But the problem is that it is substrate dependent. All these definitions depend on a specific substrate, and there is lots of blur in the understanding of this notion in literature and so on. It is very well sorted in each field, but there is no bridge in between all of them, and we have no systematic theory of self-replicating systems. So that's why I decided to build one. So we start by defining what a component is, because we have this big state with different components in it, and we need to separate things that are unique and things that are connected or not, and so on. So for instance, in the hypergraph writing setting, it would be connected components, and in the string substitution systems, it will be letters, for instance. And then we create a causal descent, a causal graph from it, where any node, any component B descends from A if A gets rewritten into B. So if something creates something, it has a relation, it has an edge. And okay, if you apply this for any given substrate, any given system, you get these graphs, these type of graph. In that case, it's a tree, but it can also be a DAG if you allow for merging rules that produce less components that they have in the beginning, for instance. So this is an example of a substitution system where the components are letters, and we can track that here A gets rewritten into A and B. And for instance, this B was produced by this A at this given point, where this B was produced by the first rewriting rule and by the first A. So you can see that there is an actual causal structure of the rewriting rules over the components we have. And this is extremely important in my opinion. Then you have other examples. You can have merging. So this was splitting. You have one component that's split into two components. And this is neither splitting or merging. This is just static. In that case, you have merging, which means that two components are combining into another. This is just an example again. Then we can have more complex structures. We'll talk about this later. But no, okay. You surely noticed that there were colors on my graphs, and I did not explain what this is. It's called an equivalence class, and it's defined as such. We need to know what does equivalence means in the specific substrate. So for instance, in string substitution, it's just equivalence between characters. In cellular automata, it would be pattern equivalence, pattern equality, and but we allow for rotation and translation, for instance. And for hypergraph writing, it would be isomorphism or something. So this is strict equivalence between two components, but then we can build lineage of these components, and we restrict it to only chain lineages, which means the part of the lineage where the component doesn't split or merge. So it just grows quietly on itself. And these are called chain lineages. And if you merge all the chain lineages that share the exact same node by the equivalence you provided, you get families or class equivalences. I'm sure you're a bit confused, so I'm going to explain it with an example. It's easier to understand. So you've got this graph that comes from this string substitution system. The first thing we can identify very easily is that the x is alone. The x is always an x. It stays like this, and it's always isolated. It doesn't split and doesn't merge. It doesn't do anything. It just stays like it is. So it will be one equivalence class. Let's call it blue. Then we got, let's look at the bees. So the bees, will have 4 of them, and every one of them is growing into a C, which means that they are not exactly different. Because if every bee grows into C, we can see that it's just, C is just a grown-up version of B. So we can group them into the same equivalence class. And we can do the same for A ultimately, because A is alone, but it also grows into B. So we can merge everything here in the same class and call it the red class. And eventually you've got this graph with the colors. You have 2 equivalence classes, and it makes sense because the first one is producing X's and it's repeating itself. And also there's no replicating events that occurs between A, B, and C. So everything is happening in between the classes, not inside of it. So, okay, this is the entire model. There's not much than this. Then after that, we need to think about the model and to explore the state space and understand what it means. So let's have a look at some examples. There is what I call a residue. So it's a component that doesn't evolve. It just stays as it is every time. So it's just a chain with every node being equivalent to itself, to the previous one. Then we have a chain that is not a residue. So this one evolves, but doesn't split or merge. It's not very interesting. For instance, gliders in the game of life are such structures because they doesn't split. They don't split. They just glide on the plane, and so they go through different states, but they don't give rise to new classes. And then we have more complex structures. So this is called a residue gun, because you have this red structure that replicates itself, and next to that you've got these. It's also producing chains which are residues. It doesn't change. It just stays the same. So A is copying itself through the evolution of the system and also producing some Bs. And here you have the same idea, but it's not a residue gun, it's a chain gun, because here the blue thing is actually growing, so it's evolving. You've got this this evolution. It's not exactly the same between BBB and B, so you've got, that's why we call it a chain. And then here's another example of chain gun in the hypergraph writing setting. You can see that the red class is producing circles that grow into bigger circles and so on and so forth. Here's another example of merging, another one where 2 different classes are merging into one single one. This is an example of cross merging where you have X and A that recombines into X and A again and again. It's just an example of what could happen in the state of space. And then, okay, what does that mean for self-replication? Because it's cool to have this graph layout, but we would like to understand what does it mean and how is it useful to understand self-replication. So we can define a replication path as a path between 2 nodes of the same color or the same class that do not include any node of the same color inside of the path. So for instance, in that case, every path is the same because in 2 nodes, every edge is a path. It's a minimal path between red and red, and you could not extend it to AAA because it would have a red node inside of the path, and this is not allowed. So every edge here is actually a replication path, and that is how you can define self-replication. So a system would be self-replicating if it has a replication path. But there is one extra criterion. It has to split at least once. because otherwise you just get a chain and you allow this to be replicating. So it has to split at least once, and then you can allow for merging everything. It can split any amount of time, but it has to split at least once. And then what's happening? The problem is that with that definition, you allow for constant replicators, which are replicators that eventually merge into themselves. And at the end, the population is constant. So in that case, you have A that replicates into A, but a single one. So in the end, the overall population of nodes stays on average exactly the same in constant. So to get rid of this problem, you just invoke a new property that is proliferation property. And it says that a class proliferates only if it produces at least two descendants that are never going to merge together. That prevents you from having the previous problem. And so you can use this as a new filter on self-replicating systems in order to restrict the space of what you're searching for. And then there is one of the best contribution of the paper, to my opinion, which is mediated self-replication. It's a new concept. The idea is that some replication paths have foreign nodes in it. So for instance, here, the red class is replicating into a red again, here and here, but it has to go through blue dots. And this is important because in real life, and when we're trying to find, the origins of life or things related to biochemistry or biology, you always have mediated replication. And it's usually not just something cloning itself again and again. It has to go to several steps and to, mature and split and grow and merge again and so on. So this is very useful to describe it. And also the great thing about this property is that it's graded. You can quantify it by using the splitting distance and the merging distance. So on a path, every merging edge can be counted and every splitting edge can be counted. So in that case, the splitting distance, the minimal splitting distance for the class red is 2 because it splits 2 times before recovering the red, right? And this is just a representation of the rules in an abstract way, because we don't care about the substrate in that case. It's just about the replication that's going on and the underlying process. And then, okay, we have this system, we have the rules, we have the properties, everything works. Now let's explore the substrates, because we have different substrates. So for the hypergraph setting, one rule can give rise to several small rules, which would be atomistic rules, exactly the same kind of rules we have in the substitution systems. So in that case, you have 4 different rules and 4 different classes of equivalence, which are red, blue, yellow, and green. Green is just over there before the split of 2 that leads to 2 blue dots, if you do not see it. And so this is on hypergraphs. It produces this, which is a set of, splitted different hypergraphs of different sizes. And every different shape of these graphs corresponds to a specific class of equivalence, as you can guess. And it has been run for 50 steps. This is another, this is another way of representing data. It's just that some graphs get very complicated, and to extract the structure of it, we can compress all the big chains of a single color in the left-hand side graph into a single node on the right representation, so that it's easier to see the structure of what is splitting, what is merging, what is happening in the system. Because otherwise you can get very easily distracted by these long columns of the same color, and essentially carries exactly the same information, but you need to be aware that the time is, so on the left-hand side, the time is flowing from top to bottom linearly. Every layer corresponds to an actual rewriting step, but on the right-hand side, the time is not flowing from top to bottom exactly, because some nodes can appear from nowhere, and all the leaves are actually at the very bottom of the extended linear graph. Then you have another example of self-replicating mediated system in the string setting with a reduced version. You can see that it has merging as well, not only splitting in that case. So it is an example of splitting and merging replicator. And then you have more complex ones. This is in the smallest glider gun we have in the game of life, which is called the Gosper gun, if I'm not wrong. And it is huge because the cellular automata, the 2D cellular automata, you have to account for all the grid at the same time in each rewrite step. That's why the reduce graph is so big. But you can see the two horizontal lines that are corresponding to two gliders that are ejected from the glider gun. Then you've got, okay, fine. So this is an overview of what's going on in the computational substrates. But now what about life? Because that was the original question. Actually, we can use a rewriting system to describe organic chemistry, and especially the chemical reactions that could be happening in the early days of life. In that case, we're going to define an initial condition, which is at the very bottom of the screen, and then a reaction, this one, that is described by this chemical formula. And this can be essentially applied exactly the same way as rewrite rules. The only difference is that you have to specify the order in which the things are executed, but it doesn't matter in our case. And so if you run this, you get this, which is exactly resembling what I called a two splitter or a two cloner, because it splits into two copies of itself. But because chemistry has conservation of mass, you have to have some fuel, an extra species that allows for the replication to happen. You need to have some atoms in order for the system to be able to copy itself, because otherwise it would just create matter from nowhere, and you cannot do that in chemistry. So it's a specific type of cloner that I call the fueled cloner, because it's able to clone itself, but it has to have fuel. And then, okay, this is the best result I've got so far. There is this complicated reaction in chemistry that we call the Foremost cycle. And this is one of the best results we have in biology when it comes to autocatalytic networks. So it's a multi-set of different molecules and reactions that grow together and produce copies of the initial molecule that is called Foremost. And it is actually used in some living beings for different usages. And it has been studied for many years now. And what I did was try to reproduce it in a computational way using only the Wolfram pattern reaction function, and to try to abstract it into something that is only about rewrite rules. So what I did is a subset, but the essence of the Foremost cycle, by defining three reactions. The two first ones are just elongating a carbon chain, and then the third one is splitting it in half. This is essentially what's happening in the Foremost cycle, but with more steps and more intermediate nodes and molecules. But it is exactly the same idea, and it's using actual organic chemistry substrates. And if we apply this to an initial condition that is appropriate, and we provide enough fuel, we've got exactly what we expected: a self-replicating diagram where you have the lineage graph, you've got the fuel in blue, you've got the initial glycoaldehyde in red, and it grows once, it grows twice, and then it splits into two glycoaldehydes. And if you have enough fuel again, you can do it forever and produce any amount of copies of the initial condition. So this is a proof in some sense that the substrate agnosticism I claim is also applicable to biology and to biochemistry in that case, and that it could be used for origins of life research in that case. So here is an expanded version of the same lineage tree, but here you have only one cycle where red turns into two reds. But in order for the intermediate steps to be colored, we have to run it for more time so that it detects that there is redundancy. So that's why we have yellow and green classes more than in the previous diagram. But it's exactly the same. You have more fuel, you have more reactions, but at the end you produce exactly the same, which are copies of the glycoaldehyde. So in conclusion, in essence, we provided the first substrate agnostic definition of simplification I know about. There is also a team, I need to mention it, that drafted and that developed a causal lineage framework for simplification, but it was substrate dependent and constrained to a specific cellular automata, and it was not formalized in the way I did. So I'm currently discussing with them to possibly collaborate or to have their opinion on my work. Then we also provide many metrics that allow for very easy screening and testing of simplification in any conditions on any substrate, whether it's chemistry or computational. And then a new mediated replication property that seems very central to me and that could be very useful in the next few months or years. I hope so, in the field of origins of life, if we manage to apply it to automated discoveries and to exhaustive search and so on. And then I need to mention some limitations of the model. So first, I assumed causal invariance throughout the whole work. That means that the update order actually matters. And I only selected the case where it does not, because it's way easier to define the model, but I could write a multi-way system of it that would be very hard to read and to visualize properly, but it could be doable in future works. Then I acknowledge the lack of rarity claims and radiological reports, because this would be really nice to have a quantification of how rare is self-replication or mediated self-replication or proliferation and so on in some settings, for instance, in hypergraph and string substitutions and so on. And it could give us an idea of how rare could be the images of life in organic chemistry settings, even though, you know, life is more complicated than just these abstracted reactions. And then I also acknowledge that the relevance to origins of life research still has to be explored, but I'm confident that it will find some space where it could be useful. And then the last point is just a matter of implementation. It doesn't matter. So yes, thank you so much for letting me present this work. I'm open to questions, of course, and you can contact me on any social media to discuss this subject if you're interested in this. Thank you so much.
Thank you, Matteo. So a question on the equivalence classes. So you're taking the history of this rewriting system and then color coding it based on heredity?
Heredity, yes. Yes, essentially heredity, but in that case, you need to provide a map that is substrate dependent. So when you instantiate the model, you have to decide what does causal descendance means. So heredity, as you say it. So it's very easy in most cases. In this case, you know, if B comes from the rewrite rule that comes from A, then it belongs, you know, to the descendants of A. And in most cases, it's easy to understand, but yes, in this sense, yes.
So is that what is being shown here? The different classes, or these colors here represent
yes,
the different letters?
Exactly. The colors here represent the rules that we extracted empirically, if I so to speak, from the graph. So when we look at a graph, we can see that easily that red gets rewritten into two pieces of blue, that blue gets written into red and yellow, and that yellow stays yellow. And this is essentially a kind of compression of what's going on in the graph. And the smaller the compression is, the less interesting the model is in some sense, or actually not interesting, but the less, you know, diffuse and complex it is.
Right, right. Um, so I'm
And
curious about
each,
how that translates to the chemical case.
yes,
That is, in some chemical reactions, it's pretty easy to say that those two chemicals merge to make a bigger one, where each
mhm.
piece is represented inside the bigger one, verbatim. Um, but then it's like sometimes you have two chemicals which come together and then they make two different chemicals that are
Yes.
swapping parts. And so does that
So
how
that
does
that would
hereditary?
be that would be a case like this, right?
Mm,
So you have two chemicals and they both combine into two other chemicals, and you don't know exactly which piece is where, right?
yeah.
This is it. So of yes, it's a current limitation of the model. I discussed with Bob about the subject, Robert Nachbar, for
Yeah, I
reference.
just joined. I had another meeting that I just got out of.
Oh, hello. So yeah, we discussed this, and you suggested that we can make a track. We can keep track of the provenance of each atom, basically. So we could build a specific version of this framework where you keep track of each atom so that you know exactly where did it goes.
Yeah, I mean, one would do that by using pattern reaction and apply reaction.
Yes, I'm currently using pattern reaction and apply reaction in the organic chemistry substrate, and so um, I'm just curious about the map that would track the provenance of atoms when you have merging, for instance.
You need to use, well, depending on how you write the reactions, the pattern reaction, you need to provide an atom mapping
Yes.
that says that, atom one in reactant one goes to atom J in product K.
Perhaps you can show some of those slides for Robert's sake.
Okay, so here is my favorite result and probably the most relevant for you for the conversation. So I was able to make a lineage tree out of a simplification of the former cycle using pattern reaction and applied reaction. And I was able to use exactly the same tools and abstractions as in the other substrate, the computational ones. For this one, I can apply it to cellular automata, to a string substitution system, and to any effectively any substrate that follows the rules I presented before. So basically, you need to have an equivalence map that tells you what does that mean that two objects are equivalent, and you need one component function that turns a state into a set of components. And in that case, it's just a multi-set of molecules, so it's very easy. And equivalence is just molecule equivalence. But we can push it further so that we divide each class into more equivalent classes depending on the provenance of atoms. I know that you did that in your work on multi-way systems of pattern reactions. So I would really appreciate to have a look on that and try to implement it myself.
Okay, this looks kind of like a token event graph that you're working on here. And I'm assuming these are directed edges
Yes,
that
it
are
is
flowing downward.
directed. Yes, it is. But it's not token events. It's the way. Okay.
Yeah, okay, yeah. You're just showing which two molecules join to form another one.
Yes.
Or actually on the left, it looks like you're starting with three molecules, and then at the next step you have two, or two of them have combined, and one has just been propagated, and then going to the third step, You have a single
Yes.
molecule, in the fourth step it splits.
Okay, the two sides of this are the same but not the same representation, because here you have each line, each row corresponds to one step in time,
Right.
where in the left, yeah, in the right hand side you have it's just
Yeah.
that the two of them are combined into the same node.
Right. Right. Okay.
I was wondering, Matteo, do you have a sense for the physical conditions required for self-replication foremost reaction? Do we know what are the situations in which it can occur, and how can that inform when self-replication can happen?
Are you asking for
Yes.
chemical conditions, Willem?
Yes, chemical, physical, I don't know. You tell me.
So I'm sure that Bob has probably more relevant answer than I do, but essentially these conditions, if I don't know exactly what are they, I know some of them. It's essentially resumed in that graphic. You need a lot more than what I described, but it's just for demonstration purposes. But I know that these constraints are outside of the realm of the lineage graph, in the sense that they are features of the rewriting system itself. And the goal would be to define, if you had to solve organic chemistry and to turn it in one-to-one mapping in a rewriting system, you would have to account for lots of parameters that we currently do not account for, such as pH, temperature, I don't know, lots of things. And these constraints are part of the system itself. And what I believe is that if we were able to write such a program, such a system, then my model will apply to this. But because we have not built the system yet, the best I can do is to apply it to this simplification we have. I'm also aware that there is more complete, more advanced rewriting systems for organic chemistry that exist. I'm just showing this because it's convenient, and because it shows that it can be applied everywhere. But I'm sure that it could be applied to any formal system, really, where you can, specify the equivalence and the component distinction, as I mentioned previously.
Hi, Matheo. Thanks for your talk. I'm a bit more curious about formal structure and the properties of the formal structure. And I was wondering if you have an idea of whether kind of self-replication is closed under composition if I have two systems that independently are self-replicating and they share some parts of the alphabet. Would the coexistence also be formally will be forced to be self-replicating or not?
Okay, to my current understanding, okay, there is different kinds of rewriting systems. Here, for the sake of the presentation, I simplified by just saying that everything is kind of the same, but actually there is synchronous updates, there is global rules, there is automatons with stacks and ones with no memory and so on. So if you have two different rewriting systems and you combine them together, that would mean that you create a new rewriting system, right? And that new rewriting systems will have essentially any new shape. I'm not sure that there is probably work being done on this, but there will be new properties of the system. It could be just composition of these two, or new properties that emerge from the combination of two different ones that are sufficiently, chosen in a certain way. But what I know is that as long as you're able to provide me, equivalence and distinction between components, I think I can build a lineage graph for it. But then, okay, to answer your question, you're asking me if the self-replication conditions and the shapes that would emerge, right, would be kind of comparable, or like resembling, right? Is that the correct?
No, I'm just meaning you have two systems independent. They do have the properties that you describe. Is the composition of them also having the same properties in general?
In general,
Understand there's many details and all that, but what would you expect? My question is pointing at, let's say, at the origins of life, many people, and I also think that many people think that there's a possibility that there were many systems trying to kind of kickstart self-replication at once, right? So I'm just curious if these properties are kind of contagious in some sense, formally speaking, then once one of the systems gets that property, and then there's another with the same property, instead of competing, they can just propagate it much more easier.
So it's not true in the general case. But there are some invariants that are conserved over composition of two writing systems. Again, you would have to match only systems that have kind of the same architecture, overall architecture, because if you combine one, I'm sure you could do it in some sense, but okay, it will inherit from the constraints of those two, of course, obviously, but then there is no general rule of what's going to happen if you do that. For instance, the very simple case, if you get a substitution system and you combine it with something, I'm not exactly sure the shape of it, but I'm sure you could get a serial automaton, a one-line one. And I'm not sure you would get exactly the same properties as in a proper string substitution system. Again, okay, and know what's interesting is that first, two things. First, in my model, the property will remain the same everywhere. So in the case of your idea of combining those two, it will remain the same. No, the distribution of each, type of replication would probably be different or not. It depends on the combination, of course. And then another another answer I can provide you is that I think most of the rewriting systems, I'm trying to avoid bold claims. So I think that in most rewriting systems, you could extract it and reduce it to a set of atomistic rules. And these atomistic rules would be equivalent to a string substitution system. And that's important because I think, I should have to check this, but I think that even complex things like hypergraph rewriting systems and so on are reducible to string rewriting systems in some sense, because you could build a mapping to binarize the data and to discretize it into a unique and atomistic framework that has the exact same properties. But that problem is hard, and especially to get these atomistic rules only by looking at the big one, the big picture, is very hard. And I'm sure this is one of the problems that the institutes would be interested in doing, because during the Wolfram physics project, you have been thinking of how do we infer what's happening atomistically just by looking at the rule of a Wolfram model, for instance. But I think, essentially, I think that the properties of a string substitution system would be preserved when you combine those two, and that's the only claim I can make.
Oh, thanks.
My only thought towards that question, Pedro, is sort of if they have a rewriting system like that incorporates strings A and B, let's say, and then you have like B and C on the other one, so they fight on the B strings. That is, there might be some common atom or type of
Oh, so you're talking about the same substrate but different rules, and you add them up together to create a new big rule that that has
they might compete over some common resource that's integral to both systems.
Then I can provide a proper answer to this. So if we do that, at least I can think of one counterexample in which, for instance, the left rule that you added, so you have left and right system, you combine them, and the left one will dominate the whole system and clean it out so that nothing exists anymore and only the left side exists. I could probably show it later, send it back to you, but yeah, we can definitely build that. So that means that in the general case, the properties does not combine, because you can easily build one system that is dominant enough to clear the other one. And by the way, in most cases, rewriting systems are you have to define an update order in order to for my model to work. So the right hand side or the left hand side, whatever you prefer, you have to specify an order of updating. And so the model that will be on this side, the first to be applied in some sense, will win, because it can define what will the model look like after that. So if we combine left and right, and we start by left, then probably right will be, vanished.
But I imagine it also depends to some extent on the kind of algebra that you have for combining rules. Because if I take two string substitution rules, for example, and suppose that they contradict each other. So, one says A goes to B and the other one says A goes to A, or AA, for example. I could think of a few different ways of combining those rules. I could combine the rules so that, joining them basically makes the output somehow concatenated, and then I'm just imagining the way that you would choose to combine rules, whether the rules are sort of competing if they disagree, and one of them might win over the other when a particular substitution opportunity arises, or whether they're somehow flattened into a single new rule that doesn't contain any contradictions. The dynamics would obviously differ. And so, so I'm not sure whether in one case, in some cases, depending on how you set up the algebra of like how you would combine your rules, that maybe you would be able to enforce or guarantee. I mean, maybe there's some constraints that you can impose that would allow for what Pedro was discussing, where you have one rule that does with self-replication and another one that doesn't, and you combine them, and then the self-replication propagates to the combined
you could
rule,
build
for
it.
example.
You could build it very easily. The trivial example would be to have a self-replicating rule, and you append at the end one very trivial rule, such as A rewrite into A, and that works. So there is probably some examples where that works, and probably some examples where that doesn't. And to answer your what you just said, under causal invariance, because order matters, contradiction is not really contradiction, because if you have two different left-hand sides and they compete in some sense to have different right-hand sides and to turn into each other, the problem is that there is an update order that is well defined, so there is no competition. One is already winning, but it doesn't mean that the other one will just get removed, because in some cases the first rewrite rule could cause some changes that are catched by the second one, and so on.
Phileas's remarks about combining rules together, the left-hand sides are the same, but the right-hand sides are different. It seems to me it would be very arbitrary how the combination would occur. You had mentioned an algebra for it, but I don't know if there is one that's well defined already that would take care of that. The other thing that I'm having a really hard time following is competition here. And what Matteo said, I think I heard him say that if the left-hand rules are always applied before the right-hand rules, they will tend to dominate. But I still don't see how they would be competing, how you would end up with a final rule set that didn't have anything from the right-hand side in it.
It depends if they are orthogonal, for example. So if you have one system that applies on a specific alphabet, and the other system that you're combining it with that applies on a totally different alphabet, then there's no competition. But if you have the same alphabet for both, and then you apply only you apply first the left hand side, the left system, and then the right system, of course that it will look more like the left system because it was applied first, so it
Right.
drives the shape of the entire thing.
Right, but in the end, you still have a system of both sets of rules, and your super rule that says the rules from the left set get applied before the right set. And Stephen has a post from a couple of years ago where one would try applying the rules in all possible orders to come up with a multi-way system for the rewriting. And again, I think your imposition of the rule, the super rule that left-hand rules are applied before right-hand rules, is in a sense kind of arbitrary.
Yes, it is. Exactly. That's what I try. Yeah. And especially it doesn't really matter because in real life, okay, it is certainly ordered. There is certainly an order of how reactions are applied, but it doesn't matter on in which it doesn't only depend on the molecules that are there. It matters on the conditions of the environment first, and of course one reaction can modify the conditions of the environment and provide new substrate so that there is a continuity and an order in the reactions, but order of the updating, the way it works in causal invariance or causal variance in that case, is not really relevant to chemistry. That's why I guess that's why you do multi-way systems.
Well, okay, now somehow we've slipped from string rewriting systems back to molecules, so I am really getting confused about the logic here.
Because what I'm saying is that everything is equivalent under lineage graphs. The principle is exactly the same. If I replaced this graph by exactly the same, okay, let me show you.
Your screen has not updated in quite a while, so maybe I'm not seeing what you're referring to.
No, I think it's fine.
Okay.
Do you see?
Yes, I
If
see.
If I do that, I'm able to turn this into a set of strings of abstracted rules, and actually this is exactly equivalent to the three molecule reactions. So you can see that blue turns into red. No, blue plus red turns into yellow. Yellow plus blue turns into green, and green splits into two instances of red. And so this means that these are exactly the same structure, or at least replication structure, than a string substitution system that would look like A plus B turns into C, and then A plus C turns into D, and D turns into A plus A, or something like this.
Mm-hmm.
So really, you have this strong connection between all the rewriting systems, that they are essentially doing the same thing, but with different representation, different abstractions for it, different levels of description, and so on.
I had another question, but Dugan, how tight should we be on time? Should we stop at 1 or?
We can go over.
Okay, well, I was just wondering, you did a reduction of the chains in what you're showing here, but have you ever thought about doing a reduction of something else? So for example, in the merging and branching plot that you showed, there was some, and maybe you could pull that slide up, there was some repeated like motif. So you could think about reducing it according to not
This
just the
one.
chains, it's before this, it's like just showing the merging and branching one. Yeah. Yeah, so like you can
This one?
imagine one more to the one more. So okay, I lost it now. It's like A goes to AB, Yeah, this one. A goes to BB, BB goes to A. So
Okay.
you can imagine compressing the whole A, B, B, A thing into one thing, even though it's branching and merging, and think about it like that. I don't know. What have you thought about that?
I'm not sure. Okay, compressing. Actually, this is what the rules does, like the grammar plots. So I'm not sure if I understand correctly your question.
Mhm.
So, for instance, this is a compression, like the minimal compression, in fact, that we could have for the system. Oh, wait, it's not
Right.
right. Yeah, we use the same name again. Yeah, this is the minimal compression.
Right. But here,
But it's the compression for the whole system, right? As opposed to, I think, Willem, what you're gesturing at is a compression for a particular pattern of rewritings.
yes, like so you're equivalencing here just B, the B's and the A's, but you could imagine equivalencing the branching thing, like A, B, B is one thing. We can call it some class.
Oh, okay. So
Yeah.
this would
And
be
that part
related.
repeats.
This would be related, barely related to Nora's work on mesoscale descriptions of phenotypes and rewrite rules, right? Pedro, tell me if I'm right.
Maybe,
think-
but I think this is probably easier if you, let's say, if you have an arbitrary graph and you decompose it into simplicial complexities, for instance, or whatever hyper edge. Like I think what Willem is saying, if I call this thing whatever star, then in some sense I can represent this whole thing as the star goes to star, something like that. So I think this could be useful for some cases, but it might obscure some other things that are relevant. And I'm not sure what's the trade-off between what it gives you, what new insight it could give you versus what can it hide, right? So I'm not sure. So I don't know if you think could be interesting.
But if you have, okay, you're okay, then you would be looking at invariance in the topology of the lineage graph. Oh, the lineage graph. But you would be excluding the information of the classes. Or if you don't do it, then you're just describing, actually it would be exactly the same level, to my opinion, to the grammar plot thing. Because, okay, for instance, if you've got one system that splits again and again, and okay, actually let's have a look at this. So, okay, for instance, this one, even though it's not the best, wait, I should have one that's better. Yes, this one. Okay, great. So you have, this is a replicator that's very simple. So you have A that goes into BB and so on. And so in that case, you would take this, the BBA and the ABB, and that would be the building blocks of the graph, right? The representation, the compression. But then this representation doesn't say anything more than the grammar plot, because it's essentially exactly the same piece of information. Because okay, in that case,
it might be, and also it might be harder to see any of the, because each of these shapes would have two or more or just one equivalence class, and that is important to visualize, I think. So maybe it's better to actually just have it as is.
And especially in the case of the star, in the case of the star, or the diamond, let's say you have actually one triangle, that's one rule in the grammar plot representation, and then you have the merging, that's another rule. And so if you represent both of them as compacted inside one diamond, then you could be missing something about the connection from the bottom node to another place where it does the same thing, but not for the same reasons. In that case, we have, okay, wait, I have one example of this just over there. Yes, so here I think, if I'm not wrong, that the blue dots, okay, no, not in that example, but at some point you could have one system that uses one color at two different states of the replicating system for different reasons and from a different lineage, from a different background. And you would lose it if you compress only on shapes. You would lose the connection between the colors in different shapes in that case. I don't know if it makes sense.
Yeah, that's interesting. I mean, I can say I was just imagining, like, for example, a biological cell has many branchings and mergings, but we can somehow recognize it still as the same thing when it replicates. So I was wondering about expanding your definition of equivalence classes to try to capture that. But yes, I think I don't know. I'd have to think about it more.
For that, it seems like you'd have to identify equivalence classes based on like a pattern recognition.
Yes, but I think it's a totally different work because, okay, in the case of a cell, you could model it using this, but it would not be very accurate because it's very messy. A cell is very messy, you know, thing. It has more mutations that you could even imagine, and especially if you try to take the, let's say, the 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 chemical signature or like the chemical network of an entire living cell, and you try to abstract some patterns in it, it will not be patterns of replication. It would probably be patterns of, what reaction goes into what reaction and so on. That's also an extremely interesting question, but I feel like it's the only connection is the study of, shapes on graphs or on DAGs in that case. But yes, but I'm sure we could use similar, similar tools to analyze both. And probably that reproduction, we could benefit from every work that has been done on phylogenetic trees or metabolism networks and so on, because they have developed some tools to analyze networks and to visualize the correspondences and so on.
Let's see, we have a question from the live stream. John Johnson is asking, could two rewriting systems generate the same sequence of observable states but have different lineage graphs, such that one would qualify as self-replication and the other would not?
Same sequence of observable, no, because you can define a system. Just if you look at the evolution of a system and it's exactly the same as the other one, if that's what you're saying, then both the systems are bi-simulating each other and they are the same, even though they are not defined in the same way. And okay, then if you're talking about the end state only, then yes, I believe that you can, I'm actually sure that you can build two different rewriting systems that end up having the exact same state at the end. So that would be the observable state. That would not be a sequence of states. Also, we could build a system that has the exact same sequence of, let's say, last 5 states. That is the same, but the two systems are totally different. But if you cut everything at this section that is exactly the same, then the rewriting system that is describing this section would be the same. It's just that the pre-setting section would be different, but that's cheating.
Does the self-replication by your metrics have to like Like something has to stay replicated from one step to the next, or can it go through like a sequence of steps and then arise again, for instance?
This is mediated replication, if I understood the question correctly. So, oops, sorry. Mediated replication stands for when you have a system that replicates in a mediated manner, which means that it goes through different states, which can also be self-replicating, and then only after that it comes back to its original state and start again.
Right,
Is
right,
that-
yeah. So is this replication where A goes to B, B, and then one of the B's goes back to A, and so A gets replicated, but
yes,
in a mediated
in
state.
some sense. But you have what's important here is that there is this thing C, and if you remove this thing, it's just a chain. But in that case, it's not, because you also produce some new stuff, which is yellow in that case. And this is important because it matters in the definition that it has to split. Everything has to split. And otherwise, these nodes would be the same color because they would be in the same chain lineage, according to the definition.
Mhm.
So it'd be just a single self-replication that is not mediated.
Very cool.
Thank you so much.
All right, I think we should wrap up. Thank you again, Matteo, for presenting. Thanks for everyone with their questions on the live stream. We look forward to more Institute live streams soon. Thanks once again.
Thank you so much, everyone.